Right, good morning. So, one who is joining online, can you hear me? Am I audible to you?
Yes, ma'am.
Right. So first, we'll start from topic one, right? That is molecules, transport, and health. So, before that, can I know about you? So, what is your name?
I'm fine, Bianca.
Yes, Savindi, just tell me about you also, little bit. Yeah, yeah.
I'm from Goa.
Right. Then what are you doing now? OA levels, O levels, after A levels?
So,
After O levels, right. So, I think you all have done this local syllabus, right?
yeah,
Okay, so then first we'll start the lesson, that is molecules, transport, and health. Right here, you have to learn
okay.
mainly the chemistry. Okay, so we'll see what you have to learn here. Right, the chemistry of water. So, what do you know about water? Do you know what are the that is there in water?
Molecule,
Yes, but right, just tell me what are the bonds that you can find in water molecule? Do you have any idea of that? Right now, here they are saying
yeah, oxygen and hydrogen atoms.
yes, oxygen and two hydrogen. Right. So all the reactions in the living cells take place in water, right? So for all the living cell activities, it needs water. That means the medium should be water, right? So without water, substances could not move around the body. Right now, if you don't find this water, can these reactions happen? And can this transportation take place in the body? No, it is impossible, right? So water is one of the reactant in the process of photosynthesis. I think you all have heard about the photosynthesis as well. Can you remember what is photosynthesis?
Yeah,
Any
ma'am.
idea? Yes. What is photosynthesis? Yes, just I'm not asking the definition, right? Just an idea. Have you heard about photosynthesis before? So what is it?
Yeah, photosynthesis is the process where plants use sunlight, carbon dioxide, and water to make glucose and oxygen.
Yes, very good, right? So there you use what water also for the photosynthesis it needs, right? So first we'll see what is the structure of water, right? That means how this water molecule is made up of, right? So molecular formula. First you have to know, I think in chemistry also you are studying these molecular formulas and all, right? So molecular formula is H2O. That means what? There is two hydrogen atoms and only one oxygen atom. So what is the bond that is there? What is the bond? In water molecule, what is the bond?
Polar covalent bonds.
Yes. Yes, we have started the lesson, right? But still we are in first slide. So can I know your names? Hmm? Kya? You? So you have after O levels or after A levels? All levels. Then local syllabus. You? Right. So we have started the lesson first one, right? The topic first topic, that is chemistry of water. So the structure of water mainly H2O, right? The molecular formula is H2O. That means there is two hydrogen atoms and one oxygen atom. So what is the bond that is between this molecule? What is the main bond? Read it and tell me. Have you heard about the covalent bonds? Have you heard about the covalent bonds? Yes or no? Just tell me. No. No, yes. Yes, Savindi, have you heard about covalent bonds? Yes, what is a covalent bond?
Type
Yes,
of a chemical bond where two atoms share one common pair of electrons.
yes, there is two atoms which is going to share the electrons, right? So then it forms a covalent bonds. Now, I think in chemistry you are going to study these covalent bonds, ionic bonds, and all, right? So it will not be a problem. So then polarity. What is this polarity? What is a polar molecule? Why you call water as a polar molecule? Because this oxygen is more electronegative than hydrogen, right? Oxygen is electronegative, hydrogen is electropositive. So creating partial negative charge near the oxygen atom and partial positive charge near the hydrogen atom. So if you consider about this structure of water molecule, it is like this, right? So you have to know how to draw this one. In question paper, some of the examination papers, they have asked about the structure. You have to draw it and show, right? So in that, you have to show the electronegativity as well, right? Oxygen is electronegative, hydrogen is electropositive. Got it? And the bond also, sorry, the angle, that is 104.5, right? So just remember this diagram, it is very important. When they're asking about the structure, you have to know this one. So what is polar? Why you call water as polar? Polar molecule. Read it and just try to give the answers, right? Read the part, try to give the answers, then you can improve yourself more. Got it? Now, water molecule is a covalent bond. So it is going to share the electrons. Now, why it is called polar? Yes, Avindi?
One side of molecule is silently positive and the other side is silently negative. That's the right thing.
Yes, then what happens?
It has distribution of electric charge.
What? Yes. Now this water is polar molecule. Why we call water as a polar molecule? Because oxygen is electronegative and hydrogen is electropositive, right? So this oxygen is going to attract electrons towards that. So it should be the partial electronegativity there, right? So thereby we call this molecule as a polar molecule. Got it? And then oxygen is more electronegative than the hydrogen. So than hydrogen, oxygen is more electronegative. Then what happens? It is going to attract the electrons towards that, right? In the covalent bond. So it makes covalent bond more strong. Yes. Right. So did you understand what is this water molecule and all? Any questions to ask? Did you understand the structure?
Yeah.
Right. In water molecule, oxygen pulls the shared electrons closer to itself. Why it is going to pull the electrons towards itself? Because it is electronegative, right? That is the reason. So it is going to create this partial electronegativity, right? Partial negative charge on the oxygen atom. So therefore, I'm telling you, when you are writing when you are drawing the structure of the water molecule, you have to write like this. The charges you have to mention, right? Without mentioning the charges, the structure is wrong, right? So it is known as a dipole, right? Just I will give you 5 minutes. Just draw the structure like this, water molecule. I'm not going to give the notes for you, right? So I'm going to give the presentation. After completion, you have to, I will deal with this one to you, right, in the group. So you can refer it, right? If there is important notes, I'm going to give you. This dipole, you should write out. Just draw the structure of the dipole quickly, right? Finish drawing. Right. So as a result, the hydrogen atoms which have electrons pulled away from them carry a partial positive charge. Now, hydrogen atoms will get a partial positive charge because oxygen is high electronegative. So therefore, it is going to pull the electrons towards that. So what happens? Hydrogen becomes slightly positive, right? So this distribution of charges, it is going to create a dipole with oxygen having a slight negative charge and hydrogen having slight positive charge. So in this structure, oxygen is slightly electronegative and hydrogen is electropositive, right? And it is also having that bent structure, right? So due to the change or due to the difference in this electronegativity, it is having that bent structure. Understood? Right. So if I'm asking one question from you, uh, yes, why this water molecule is called the polar molecule? Why this water molecule is called a polar molecule? Yes. Why it is called a polar molecule? No idea? Just give me any idea that you have, right? It can be wrong, it can be right. No problem. Just tell me. Yes, Savindi? Can you understand that there is an difference in electronegativity here? Can you understand that point? Right, oxygen is much more electronegative. Yes, Savindi? Yes, do you have any answer?
Electronegativity different.
What?
Electronegativity different.
Yes. There is some difference in the electronegativity, right? That's why the molecule is polar. Yes, you just write down that one. Why water molecule is called polar? Why water molecule is called polar? Water molecule is called polar due to the difference in electronegativity. Due to the difference in electronegativity, right? Difference in electronegativity. That means oxygen is more electronegative than hydrogen. Oxygen is more electronegative than hydrogen. So what happens? Oxygen is going to pull the shared electrons, right? Oxygen is more electronegative. Oxygen is more electronegative. Why is called polar? Due to the difference in electronegativity. Right? Due to the difference in electronegativity. Electronegativity is like yes, don't keep the space. Don't keep space. Electronegativity. Right. So oxygen pulls the shared electrons. Oxygen is going to pull the shared electrons closer to itself. Closer to itself in the covalent OH bond. O dash H, right? OH bond. Covalent OH bond. So as a result, you write down and on please the shared electrons closer to itself, right? Then in the covalent OH bond, closer to itself in the covalent OH bond. Covalent OH bond. Right. So as a result, oxygen gets partial negative charge. You have to write like delta negative, right? You know that one, delta. So it will get a partial negative charge. Oxygen will get a partial negative charge, and hydrogen gets partial positive charge. Right? So this is the reason why water molecule is called polar. Right. Then now this structure is also called a dipole. This is also called a dipole. Why? Because there is a distribution of charges, electronegative and electropositive, right? There is a distribution of charges. So with oxygen having slight negative charge and hydrogen having slight positive charge, it creates dipole, right? So the dipole structure of the water is also same. You have to write that answer. Got it? The electronegativity difference. Understood? If there is any questions, it just ask me, right? I don't know whether you have understood it or not. Got it? Yes, what about Samindi?
Yes, madam. Understood.
What?
Got it, ma'am.
Right. So water molecules form hydrogen bonds with each other. Now I have asked you what are the bonds that is there in water molecule, right? One is covalent, other one is hydrogen bonds. Hydrogen bonds, right? So here they are telling hydrogen bonds with each other due to the attraction between the slightly positive hydrogen atoms, one molecule, and the slightly negative oxygen atom of another. Just see this diagram. O and H, right? You can see here OH. So that is a hydrogen bond, right? So covalent bonds, hydrogen bonds, both you can find in water molecule. Got it? Right. Then this leads to many of the water's unique properties. Many substances such as inorganic kinds. You know what is inorganic kind? You know what is an ion? No idea? So did you study these ions, molecules for chemistry? Huh? You study molecules, ions in chemistry, four levels. Yes, so then you know. Right. So what are the properties of water? Can you just tell me what are the properties that you can find in water? Now this is very important, right? Just write down the heading as dipole. Structure of water, dipole. Right. What are the importance of water? Importance of water? Yes. Importance of water. Right. Water is a polar solvent. Now you know what is polar solvent. What is solvent? Now, as an example, I am telling you, I am going to dissolve salt in water. Right? So, what is the solute and what is the solvent there? I am going to dissolve uh, salt in water. What is the solvent and what is the solute? Yes. Don't be afraid, right? Why are you just like staring like very much be afraid and all? Stressful? Why? Hmm? Why is that? Just think, this is the beaker and water, and I'm going to add salt, right? So what is the solute and what is the solvent? Easy question I'm asking. Just think of your tea in morning, right? You are going to add milk powder to water. What is the solute and what is the solvent? Yes. Yes. Why you are taking so much time to answer the question? Right? This is very simple question. Don't be afraid, right? I like if you can interact with me when I'm studying. So it will not be boring as this is biology. Right? So if I'm going to uh, speak like a parrot, then you will feel boring. Right? Lecture. Right. First, as the importance, it is saying that it is a polar solvent. So many of these ionic substances like sodium chloride. What is sodium chloride? NaCl. What is it? Huh? Salt. That is salt. Will dissolve in it. So many of these covalently bonded substances are also polar. Right? This is sodium chloride is an ionic substances as well as ionic substance as well as it's an ionic compound. Right? So what happens? It is going to dissolve in water as it is also covalently bonded, right? So therefore it is going to dissolve in water. So that's why it is saying water is a polar solvent, so it can dissolve the other solutes if it is covalently bonded, right? So that is one importance of water. Understood? Right. And but not in all of the covalently bonded substances. Now, if you consider about ethanol, right? Ethanol is not going to dissolve in water. So that is an exception. Not all covalently bonded substances are going to dissolve in water, right? There are some exceptions as well, right? Water also carries other substances like starch. As a result, most of these chemical reactions within cells occur in water in an aqueous solution. So why you are calling that all the reactions that is there in a living cell is taking place in an aqueous medium? As an example, they are telling about the starch. What is starch? Hmm? What? Hmm? Huh? Yes. What else? Starch is a carbohydrate, right? It's a carbohydrate, right? And the other importance of water is like water is an excellent transport medium. Why you call water as an excellent transport medium? Why it is an excellent transport medium? Yes. Why water is called an excellent transport medium? Now all the reactions, if it is taking place in the body in an aqueous medium, right? That means in a watery medium. So why you call water to be an excellent transport medium? Because of the, read the sentence.
Dipole nature of water enables many different substances to dissolve.
Yes, dipole nature of the water. That means there is a difference in electronegativity. So that is the reason why it is an excellent transport medium, right? So this is an picture. Do you have that student book with you? Not yet.
Yes, madam.
You have. Right. This I think I have taken this one from the student book, right? Just see how the bonds are there in water, right? Now here the second diagram, this one, right? It shows the sodium chloride. So the bonds that is there, right? Covalent bonds, ionic bonds, right? You can see that one. And when water is going to dissolve in this, when sodium chloride is going to dissolve in water, this is the way how it is going to arrange, right? Now you can see chloride Cl minus. So that is what? It is a negative charge. What about this pink one, red one? What is it? Yes, hydrogen. So this is hydrogen. That's why, yes, this is oxygen. This is hydrogen. Hydrogen is positive. That's why it is close to this one, right? Negative, positive. Got it? And if it is a sodium ion, what happens? Sodium is positive. What is
Positive.
this? Yes, what is this? Huh? Oxygen. Oxygen is what? Negatively charged. So negatively charged, it's going to attract the sodium positive sodium, right? So this is the way how is going to arrange in water when you dissolve sodium chloride, right? And can you just tell me what are the importance of water? The importance, just try to answer the question. Just think what are the importance of water. Now you are taking water every time for bathing, for eating, and for everything. So what is the importance of water? Can you just tell me? Think and tell.
Helps in metabolic reaction.
Yes, in metabolic reactions. Good. What else? What else?
Regulates temperature.
Yes, temperature regulation. Then,
Universal solvent.
huh? What? Yes, solvent. That is correct.
Mm,
Yes, others. Others. So what is the school? Huh? Homa. You? Lyceum. You? Devibal. Right. So you got your results? Not yet. So when you completed this oils? March. March. Still you didn't get. When you will have it? This month. Yeah, you are supposed to do it in local sorry, Edexcel. No. I'm asking about ALs. You Edexcel. Yes. Right. Okay. So the third point is, as water is going to cool to 4 degrees Celsius, it is going to reach its maximum density. Right? What is that? Now, you know that there is a density for water. You know what is density? Density? Have you heard the word? Yes. So density is normally, there are different, now if you consider about the different liquids, they have different densities, right? For water also, there is a different density. Oil, there is a different density, right? Ganapve. Got it? So, water is going to reach its maximum density when it is going to cool to 4 degrees Celsius, right? That means at 4 degrees Celsius, the water has the maximum density. Understood that point? Right. So, if it is going to cool further, what happens? Molecules are going to widely spaced. So that's why, have you heard this ice and all will float on the water surface? Have you seen it? Ice is going to float on the surface of water. Why is that? Why is that? Ice is going to float on water. Why is that? See, this is a lake, right? This is a lake, and here you can find water, right? Now I told you that water reaches maximum density at 4 degrees Celsius. When it is going to cool more, what happens? The molecule, water molecule, the space becomes more, right? Understood? The space between these molecules become more. So therefore, what happens on the surface, ice layer will form. But can you see this ice layer on the below of this one? So that's why water is going to flow, right? Why water is going? Sorry, ice. Ice is going to float on water. So why this ice is going to float on water? Because the density is low. Density is low, right? So it forms an insulating layer and helps to prevent the water underneath it from freezing. That means normally in these very cold climates, you can find these ice layers on top of the lakes, right? But you cannot find the ice layer at the bottom. So then organisms can live in that water, right? So all the lake, the whole lake is not going to become ice, only the top layers. Understood? So this property enables the organisms to live in water even in the countries where it is cold. It gets cold enough to freeze in winter. Normally in winter seasons, you can find this one, right? I think have seen this in movies. Here you cannot find, right? Because you cannot get these winter seasons here. What about Samindi? Have you cleared this one? Do you have any questions to ask? If you have any questions, just tell me, right? So after going home, you cannot. Yes. Any question? Huh? Done. Yeah. Water is slow to absorb and release heat. It has a high specific heat capacity. The hydrogen bonds between the molecules need a lot of energy to separate them. Now here, the other importance of water is high specific heat capacity. Why is this high specific case? What is this high specific heat capacity? What is this high specific heat capacity? Don't know? Read the sentence. I want answers from everyone. Read the sentence.
Can absorb or release a lot of heat being a different temperature.
What? Yes. Yes, Savitri, I didn't get that. Read the sentence. Why you are not reading? At least read. You feel like, have you taken your breakfast and came back? Or so then you are not feeling sleepy. Right. So just read the sentence. Read the sentence. Water is slow to absorb and release heat. It has high specific heat capacity. The hydrogen bonds between the molecules need a lot of energy to separate them. Why do you call this water is having high specific heat capacity? Because a lot of energy is needed to separate these hydrogen bonds of the water molecule. Understood? So that's why we call it. It is having a high specific heat capacity. This is an importance of water. This is an importance of water. So if this is not there, if water is not having high specific heat capacity, what happens in lakes and rivers? Water is not having high. Write down the answer. Write down the question. Write down the question and try to answer it at home, right? If you don't find the answers now, just think and write. If water is not having a high specific heat capacity, what happens to the living organisms that is living in the lakes and in rivers? Write it roughly, right? I want the answer. Right. Water is a liquid. It cannot be compressed. This is an important factor in many hydraulic mechanisms in living organisms. Now, water is a liquid, right? It comes, you can see that one in liquid form. So, can you compress it? Can you compress water? Can you compress water? Hmm? No. No. This is an important factor in many hydraulic mechanisms in the living organisms. What is this hydraulic mechanisms? What is a hydraulic? Hydraulic mean? Hydraulic. Hydraulic means water. If it is operating with water, right, we called it as an hydraulic, right? Right. So hydraulic mechanisms in the organisms use fluid pressure to perform tasks like movement, maintaining structure, and other vital processes. Both the plants and animals use these mechanisms in different ways. Now, not only in these animals, plants also use the hydraulic mechanisms. Have you heard about this? Right, they have given. Can you just tell me one or two examples of the hydraulic mechanisms in plants? That means operating with the fluid pressure. Fluid, you know, right? Fluid, that one fluid, right? Fluid pressure. So, examples? In plants? Turgor pressure. Turgor pressure. What inside the plant cells keep them rigid? Now, have you heard, have you seen this stem of the plants? They will keep rigid if you are going to water the plants, right? So that is called the turgor. So the plant can maintain the rigidity. So I'm not talking about these heavy and large plants, right? The small ones. So if you are not going to water the plant, what happens? The plant will, plant will, hmm? Die, wilt. Plants will wilt, right? So if you are going to water the plants, it is going to keep its structure, but rigid. That is called the turgor pressure, right? Water inside the plant cells keep them rigid and helps plants stand upright. Stands upright. Why? Because of this turgor pressure, right? Right. When water is low, there the word is there. Plant will build. Right. The movement. Some plants like Venus, flytrap, and mimosa. Mimosa, have you heard that we're not touching the plant, it will touch me not plant? Have you seen that mimosa? Yes. So there is rapid fluid movement, close straps of fold the leaves. So therefore it can close and fold these leaves, right? Because of this turgor pressures. So water plays an important role, right? It has a high specific. Now just hear this carefully, right? I'm going to ask what are the importance of water at last. You have to keep all these things in your mind, right? Got it? Roughly, right? So in invertebrates, annelids, right? Yes, earthworms. So earthworms is going to push the body forward, right? So have you heard this? They are crawling like this, right? Why is that? Due to this fluid pressure. There is a change in fluid pressure, so they are going to crawl forward. That is also due to the fluid pressure. And what about a starfish? Have you heard about the starfish? Have you heard? No. Have you seen? No? No, yes? Yes. So they are going to extend the legs using the fluid pressure. Their legs also they are going to extend with the fluid pressure, right? So they don't have these muscles and all, right? So spiders are also like that, operating with this fluid pressure. So all these things in invertebrates. Water plays a major role, right, in this. And then vertebrates, penile erection. Here the blood is going to fill the tissue to make it rigid. It is a key function in reproduction, right? Invertebrates. And crocodiles. There is weak jaw opening muscles may assisted by the fluid pressure. Now these are just examples that they have given, right? I don't think that they have given this one or not. Just see it in a student book. And in mollusk, they are going to use this jet propulsion, expelling the water to move quickly. And then octopus, The arms are like flexible, boneless, right? So all these things are adaptations, right? Not the adaptations actually. This is operating with the fluid pressure. That means hydraulic mechanisms that is there in plant and animals, right? So that is an important feature of water, right? Then seed seed dispersal also. Plants like impatiens build pressure in seed pods that burst open to spread the seed. So in seed dispersal also sometimes the what is important? Seed dispersal, right? Right. Then the other importance is like this. Water molecules are cohesive. What is cohesion and what is adhesion? What is cohesion and what is adhesion? Water molecules are cohesive. The forces between the molecules mean they are going to stick together. This is very important for the movement of water from the roots to the leaves of the plant, right? Cohesion and adhesion. Now, here, what's cohesion? What is adhesion, right? Now, there is water in the beaker. Okay, between the water molecules, what is the force that is there? And between the water molecule and this wall of the beaker, what is the force that is acting? Can you tell me? What is the force between the water molecules? What is cohesion and what is adhesion here? Cohesion means the forces that is acting on the same molecules, right? Between the water molecules and water molecules, cohesion forces. Between the water molecules and the wall of the container, that is adhesion forces, right? So that is also one important feature of this water. And the other one is surface tension. What is this surface tension? Now here, water is acting as a stretched sheet, right? Why it is acting as a stretched sheet? Due to which force? Due to which force? What is your name? Hmm? Minaya. You? Yeah. You? Yashmi. Right. Kiara. Just tell me what is the force that is acting in the surface tension of water? Now we call that the water has a surface tension, right? So what is the main force that is acting here? Yes? Cohesion. Cohesion forces, right? So here it arises from the cohesive forces between the water molecules. So between the water molecules, there is cohesive forces. And between the water molecule and the other molecules, it is adhesion, right? Now, if you're going to drink water from a straw, what is the force that is acting? Hmm? Adhesion, right? On the wall of the straw and the water molecule. That is adhesion. Right here in the surface tension, there is cohesive forces between the water molecules at the surface. So therefore, it is acting as a stretched sheet. What is the importance of surface tension for water? Importance of surface tension for water. What is the importance? In water surface, there is cohesion forces, right? This is water surface. So between the water molecules, there is cohesion forces acting, right? There is cohesion forces acting. So therefore, you can find surface tension, right? Surface tension. What is the importance of this surface tension in water? Yes. What is the importance of surface tension in water? If there is a small animals or small insects, will they sink? No. That is the reason, right? So due to the surface tension, these insects can live, right, without sinking. Understood? Right, any questions? Any questions to ask from the part that we have completed? Yes? Any questions? No. Understood. Understood. Right. So then just tell me, Kiara, just tell me what are the importance of water? Hmm? Right. The reactions are taking place inside the water. Right. Okay. Then what else? Yes, you, Vinaya. Yes. Hmm? Temperature. Temperature as a solvent. That means temperature regulation and as a solvent. Right. Then hmm? Hmm? Organisms. Right. So that is due to surface tension, right? So what about Savindi? Just tell me one or two importance of water.
Water absorbs and absorbs a lot of heat without changing temperature quickly.
What?
And water as a minimum solvent.
Hmm. Yes, solvent. Then what else?
Water is a polar molecule.
Yes, polar molecule. That is correct. What else?
The structural support in organism.
Which one?
Water provides turbo pressure in the plant cells.
Yes, Dagger pressure. Now, yes, polar molecules. Now here, as a brief, I'm telling you, water is important as a what? So it's high specific heat capacity. It is having high specific heat capacity. So what is not going to absorb and release heat quickly, right? It needs more energy to do that. So that's why the living organisms can live inside the water. If the temperature of water is going to become more quickly, then what happens? Organisms will die, right? So that is one thing. And then cohesion forces, adhesion forces, surface tension, and then acting as a solvent, then polarity, and the dipole nature of water. So then most of the chemical reactions are taking place in an aqueous medium inside the living organisms. So likewise, if they're asking about the importance of water, you have to tell the points, right? You have to write the points like that. Okay. So then they have given one or two questions, I think. Yes. What is a dipole? And transpiration moves water and mineral ions from the roots to the leaves of the plant. Explain how transpiration depends on the dipole nature of water. Can you answer? Can you answer the question? Yes or no? Just let me know. First question, dipole? Can. Right. Answer that one. And yes, start writing. What is a dipole? Finish writing? Yeah? No? going to explain this as per dipole. What is the dipole they are asking? You have to explain this, right? Water molecule, there is oxygen is slightly negative, hydrogen is slightly positive, right? So as the electronegativity of oxygen is high, it's going to attract more electrons toward that, right? So then what happens? This will become slightly electronegative, this will become slightly electropositive. Understood? So thereby it's going to create this dipole. Understand what I have told? What about Samiti? Need to write down the answer to the dipole. Yes, Samindi, finish writing. Samindi, am I audible to you?
Yes, teacher.
Yes, what is the answer you got for this dipole?
The dipole is a molecule that has two opposite electric charges or poles.
Yes. Dipole. Now here, this is important, right? Don't try to read this. What is the dipole? It is important. So you have to know, right? Dipole. Can you see when I'm writing? Now here oxygen and hydrogen, right? It is. So this is the normal structure of both, right? Now here oxygen is more electronegative. If it is more electronegative, it is going to share more electrons, right? More electrons towards this. So therefore, this will become slightly negative, right? Partial negative, and this will become slightly positive. So there is a difference in charge, right? Always in the dipole, there is a difference in charge, right? Why there is a difference in charge? Why there is difference in charge here in this water molecule? Oxygen is slightly negative and hydrogen is slightly positive. So there is a difference in charge, right? So that is the reason why it is called a dipole. There is a difference in charge. Understood? Understood? Right now. What is the dipole? There is a difference in charge. You have to explain it, right? Oxygen is more electronegative, so therefore it is going to share more electrons towards that. Oxygen is more electronegative, so there is a difference in charge. One is oxygen is slightly negative and hydrogen is slightly positive, right? So there is opposite electrical charges. Therefore, there is opposite electrical charges. Opposite electrical charges due to the unequal sharing of electrons. Due to the unequal sharing of electrons. Unequal sharing of electrons. And there is a bent structure. Therefore, it is having a bent structure. Now, simply you just remember dipole, right? There is two poles, right? Di mean two. Why there is two poles? Why there is two poles? Because there is a difference in electrical charges. There is difference in electrical charges, right? One is slightly positive and other one is slightly negative. So there is a difference in electrical charges. So this difference in electrical charges, it creates a dipole, right? Understood now? Understood? If it is not understood, just tell me now. Got it?
Got it.
Right. So the second question is, what about Savindi? Did you do this one? Transpiration moves water and mineral ions from the roots to the leaves of the plant. Did you do that?
Not yet.
Not yet. Right. And what is transpiration? What is transpiration? Now don't think much about this dipole, right? Just remember dipole is a structure which is having two opposite charges, right? Due to the unequal sharing of electrons. One is slightly positive, other one is slightly negative. So there is a difference in electrical charges. So in your answer, there should be a difference in electrical charges, right? Therefore, there is two poles. So then it calls the dipole. So then, transpiration. Have you heard about the word transpiration? Transpiration. In law grades, I think you are studying about the transpiration. Word? No? Not right. Yes. No. Oh, yes. One or two. Yes, one or two. Huh? No, right. You? Yes, right. So why you are not answering them? I have to write like this, then you are going to answer, right? Okay. Transpiration means like this. Now in the, if you find a stone, right, and there is a tree in the plant, right, not a tree, plant like this, right, and from the leaves, what happens? Water molecules are going to evaporate, right? Have you heard from the stomata and all? You know what is a stomata? You know, right? So stomata is just like this, the structure. So from in the leaves you can find the stomata, right? So the water when it is taken from the roots, it's going to transpirate out like this through the leaves from stomata. So this is transpiration, right? Now what is the question? Transpiration moves water and mineral ions from the roots to the leaves of the plant. Now that is the definition of transpiration. It is going to move water and mineral ions towards the plant, right? So roots to the leaves. How transpiration depends on the dipole nature of water. So they are asking how this transpiration is going to depend on the dipole nature of water molecule. Right? Dipole nature of the water molecule. How it is going to depend on the dipole nature of the water molecule? Now you know dipole nature of the water molecule is like this. Right? This is delta negative, positive. Right? This is the structure. Now this is the dipole. How this transpiration is going to depend on this structure? Due to this structure, what happens? Hydrogen bonds are created, right, in between the slightly positive and slightly negative, right? Hydrogen bonds are going to create. So these hydrogen bonds helps for the cohesion forces, right? What is this cohesion forces? Forces between the water molecules, right? So due to the hydrogen bonds, it is going to create these cohesion forces. If there is cohesion forces, water molecules are going to go like this, and there is cohesion forces, and it is going to create what? Continuous water column, right? Why there is continuous water column? Due to the cohesion forces, cohesion and adhesion both, but cohesion forces mainly, right? We are talking about this dipole. So therefore, continuous water molecules, continuous water what? Column. Continuous water column is created due to the cohesion forces, right? Therefore, it is helping the transpiration. Understood how this dipole is going to help or depend on the transpiration process. Dipole is going to create the cohesion forces, and then it is going to create this continuous water column from the roots to the leaves, right? So dipole nature of what is important in that, right? That is the answer for the question. Understood? Yes or no? Just tell me. I'm going to explain it again. Yes. You? You? Yes. What about Savindi? You got the answer for the question?
Yes. Good.
Right. So this is the about the transpiration, right? Now, if they're asking about the dipole nature and the cohesion forces, you should uh, write the answer like this. That means dipole is going to create slightly positive oxygen and slightly negative hydrogen. So therefore, they can create a cohesion force, hydrogen bonds. So hydrogen bonds involved in the cohesion forces, right? Cohesion forces is going to create this continuous water column. If you want, you just write down the answer roughly.
Okay, dear.
Huh? What? Yes, Kiara, for the Edexcel in O level, right? You did. So what were the subjects? How many subjects? 7. You have for the locals 9, right? 7 subjects. All are in English medium. Single is not there. There. Right. First, you explain about the dipole, right? Dipole, that means water molecule is having slightly negative oxygen charge and hydrogen is having slightly positive charge. So it is going to create the hydrogen bonds. So hydrogen bonds helps for the cohesion forces to build up, right? These cohesion forces involved in the creating a continuous water column from roots to the leaves, from roots to the leaves, right? So thereby the dipole nature of water molecule is important in transpiration, right? You don't have this student books with you, right? Right. So you have this, Savindi, you have the student book,
Yes, teacher.
right? So the page number 7, right? You have to do all the checkpoint questions that is there, right? Page number 7. Do all the checkpoint questions. Got it? And you, I'll read this out, right? She's going to wait. You have to do all these questions as homeworks and just keep another book for homeworks and question papers, right? For questions and all, you have to keep another book. It is good if you are keeping it as CR books, and every time you have to bring it. And definitely, you have to do all these questions also, which I'm giving you, and I'm going to correct it, right? So you have to do it.
Okay, teacher.
Right. Just write down this and she will write it after that, right? So what are the differences between the first question? What are the differences between ionic substances and polar substances? What are the differences between ionic substances and polar substances? Right. Then the second question, how are hydrogen bonds formed? How are hydrogen bonds formed between water molecules? How are hydrogen bonds formed between water molecules, and what effect do they have on, what effect do they have on the properties of water? On properties of water. Do they have on properties of water? Third question, discuss how the properties of water affect living organisms. Discuss how the properties of water affect living organisms. Right, all these 3 questions you have to do at home, right? Homeworks. Right, then let's start the other part, that is carbohydrates, monosaccharides, and disaccharides. So just tell me what you know about the monosaccharides. Have you heard about this one? Monosaccharides and disaccharides? Right. So just tell me what is a monosaccharide? Heard but no idea? Right. First, we'll see what are the organic compounds, right? Now, these organic compounds and all you will learn in chemistry, right? But there is a small part here they have included in your book. So we'll see. Organic compounds are biological molecules, right? So organic compounds, they all contain carbon atoms. Now, if it is an organic compound, it should contain a carbon atom, right? But not only the carbon atoms, they are having hydrogen, oxygen, and sometimes, normally for these biological molecules, it is having CHO, right? CHO. What is the CHO? Carbon, hydrogen, and oxygen. Right. And less frequently, sometimes only, they will have the sulfur, nitrogen, and phosphorus. Right. But this is not common thing. Sometimes they will have, right? So mainly in a biological molecule, it should contain CHO, right? So each of these carbon atom can make 4 bonds. You know how many bonds that carbon can form, right? It can form 4 bonds and it can connect to 4 other atoms. It is going to connect to 4 carbon atoms. And I have given 3 questions. You just write it from the print, right? And do it in home. Right. Now here this is the carbon, right? Carbon can form 4 bonds, like, right? Carbon atom can have 4 bonds, right? That's why the structure they are telling. Carbon atoms bond particularly strongly to other carbon atoms to make long chains. The 4 bonds of a carbon atom usually form a tetrahedral shape. So this is the tetrahedral shape, right? Just see this diagram. This one, tetrahedral shape, right? So carbon compounds can be rings, branch chains, or any number of three-dimensional shapes. So in some carbon compounds, small molecules bond with many other similar units to make a very large molecule called a polymer. The ability of carbon to combine and make macromolecules is the basis of all biological molecules. Right? That means this carbon, this is the structure. 3D structure is having, right? So carbon can form any molecule like this. It can bond to the carbon like this, right? It can have hydrogen in this way. It can form long chains of carbon, right? If it is forming long chains of carbon, we call it as what? If carbon is going to form long chains, then it's a, what is it? Huh? Huh? What is this? Polymer, right? Polymer. Right. So these small molecules is known as what? Monomers. So several monomers are going to form the polymers. Right. So then the ability of carbon to combine and make macromolecules. Macromolecules mean what? Macro mean large. Micro mean small. So this is macromolecules it can form, right? The biological molecules. Right. Now just see the structure here. This is tetrahedral, right? 3D. Now here the bonds are like this. That means it is just a 3D shape they have given here, right? Now one bond is back and one bond is front, like that. That means they have to show that carbon can make how many bonds? 4 bonds. Right. Then first we'll see what is a carbohydrate. What is a carbohydrate mainly? Energy source, right? It is an energy source. Now here it is important in cells as a usable energy source and important in human foods around the world, right? For all the different types of foods, it is based on carbohydrate, right? So it is an energy source. They are also important for the storing energy and important part of the cell walls in plant, fungi, and bacteria. Have you heard about cellulose? What is it? Cellulose. What is it? What? So I can hear chiki 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 chiki. What is cellulose? Polysaccharide. What is it? Where can you find cellulose? Plants. Which part? Yes, finally. Cell walls you can find, right? Cellulose. Right. So sugars and the starch are the best known carbohydrate. So you know about the sucrose? It's a sugar, right? So this is also best known sugar and the starch mainly. White crystalline table sugar is what? That is sucrose. So glucose is used as a fuel by the cells of our bodies. Starch you can find in rice, flour, potatoes, right? So the basic structure of the carbohydrate is the same. Now, if you consider about all the carbohydrate, if it is a disaccharide, polysaccharide, a monosaccharide, anything, the basic structure is same. Made up of what? What are the elements? What are the elements? CHO, carbon, hydrogen, oxygen, right? There are 3 main groups of carbohydrate. What are the 3 main groups of carbohydrates? Monosaccharides, disaccharides, polysaccharides, right? So we'll see about the monosaccharides first. That is a simple sugar, right? Made up of what? CHO, right? The monosaccharides are simple sugars in which there is only 1 oxygen atom and 2 hydrogen atoms for each carbon atom in the molecule, right? So the general formula is written like CH2O, right? CH2O. The general formula is for monosaccharide, right? Monosaccharide is simple sugar, simple carbohydrate. Simplest, right? C, what is it? CH2O. This is the formula for monosaccharides, right? There is only one carbon, right? Right. Then this is the general formula, and CH2O. N means you can put N here, right? N can be any number. So N can be any number. Triose sugars. Now, if it is triose, just wait. Triose mean what? Triose mean 3. 3 carbon, right? Have 3 carbon atoms, and the general formula will be, you can compress it and write, right? C3H6O3. So they are important in mitochondria where the respiration process breaks down glucose into triose sugars. Now you are learning in, I think in A2 syllabus about this respiration. right? There you can find this glycolysis and all that carbohydrate glucose is going to break down into three carbon compound, that is pyruvate, right? So here you don't want that, but they are telling here for the respiration process, carbohydrate is going to break down into triose sugar, right? So that is triose. If it is pentose, pentose mean what? 5 carbon. So they are having 5 carbon atoms, and general formula you can write C5H10 and O5. So ribose and deoxyribose are the important nucleic acids. Nucleic acids you are going to learn, right, in proteins. So then you can understand what is this ribose and what is this deoxyribose and everything, right, in DNA and RNA. So which is going to make up the genetic material. In hexose sugars, 6. Hexose means 6, right? They are having 6 carbon atoms. General formula is C6H12O6. So these are the best known monosaccharides, taste sweet and include glucose, galactose and fructose, right? Have you heard about this glucose, galactose and fructose? No? Yes or no? Yes. So these are monosaccharides. Right, this is, they have given here a ribose, right? Picture. Right, here about the glucose, right? So there is 2 different types of glucose, right? One is alpha glucose and the other one is beta glucose. So what is the formula of glucose? Glucose C6H12O, C6H12O6, right? So alpha glucose and the beta glucose are 2 isomers of the sugar glucose. What are isomers? Write down the question, what are isomers? Homework. This is chemistry question. So which differ in the orientation of the hydroxyl groups, right? So why they are different? Why these isomers are different? Because the orientation of the hydroxyl groups is different, right? That is the reason. So here they are telling OH attached to the first carbon atom, which is known as the anomeric carbon in their ring structure. So both the isomers of the glucose, meaning they have the same chemical formula, but their structure is different. Why their structure is different? They are having the same formula, but the structure is different. Why? Why the structure is different? Orientations are different. The hydroxyl groups that is attached is different, right? The orientations. The attachment is different, but the chemical formula is same. So that's why they are called the isomers, right? So alpha glucose. Now just hear this one carefully, right? What is the difference between this alpha glucose and beta glucose? The molecule is same, the chemical formula is same, but the orientations, the attachment of the hydroxyl group is different. Now, if in high alpha glucose, where can you find the hydroxyl group? Where can you find the hydroxyl group? In alpha glucose, hydroxyl on carbon 1 is positioned below the plane of the ring, right? In alpha glucose, just see the structure here. Alpha glucose, where can you find the attachment of the OH? Right? And just in beta glucose, hydroxyl group on carbon 1 is positioned above the plane. Can you see the difference here? Can you see OH in the first carbon, that is first carbon with the anomeric carbon, right? Hydroxyl group in the first carbon is positioned below the plane. What about this? Hydroxyl group is positioned above the plane. So that is the difference between these two isomers, right? So this is also very important, alpha glucose and beta glucose. You have to know what is the difference between these two. Got it? One is hydroxyl group is positioned below the plane. Other one is positioned above the plane, right? So, right. If not, you can tell like that on the same side of the CH2OH group, right? OH is attached on the opposite side here in the alpha glucose. OH is attached on the same side of the CH2OH group in beta glucose, right? That is the difference. Right. Then disaccharides, the double sugars. What about disaccharides? How a disaccharide is formed? Yes, Savindhi, did you understand what is a monosaccharide and all? Any questions to ask?
Yes, teacher, I got it.
Right. So then what is a disaccharide? Mono, di. What is the difference? Hmm? Right, it's 11:26, right? Just write down this question. You are having the afternoon class also today? Yes. Right. Write down the question. What are carbohydrates? What are carbohydrates? Hello? Uh, uh, ක්ලිනික් එක නම් තියෙන්නේ මොරටුවේ මම මේ දැන් නම් චුට්ටක් බිසි හවසට චුට්ටක් ගන්නවද after 5 වගේ හරි just write down the answer. What are the what was the question? Yes, what are carbohydrates? Just write down the question answer. If you want, I will put this slide, right? Because I'm giving the notes. Now, I will give you today the presentation, right? But after going home, you have to take the printouts of this, right? And then highlight all the important things and you have to study, right? After completion of the chapter, I'm going to give you a revision paper. You have to answer that, right? That's why I'm telling you, you have to study, okay? From the first day. If not, it is very difficult for you because every time when you are studying, when I'm teaching, everything is adding up, right? So it will be easy for you if you're going to study from today, right? So this is the first slide. If you want, you can read this one and write down the answer. After writing, you can take the interval. Done? You have to write carbohydrate as a biological molecule and it has CHO. So that is the most important part. CHO, right? Yes, this is the most important part. When you are writing down, when you are writing down the answer, you should include about the CHO mainly, right? So they are asking about the carbohydrates. So carbohydrates, it can be monosaccharides, okay, I didn't heard the answer of Savindi. Yes, tell me what is your answer? Yes, Savindi.
Carbohydrates are organic molecule made of carbon, hydrogen, oxygen. Carbohydrates are um,
What?
I'm
Carbohydrates.
still writing.
That's all.
Yeah.
Right. Carbohydrate. They're asking about the carbohydrates. Then you should write about it as an organic molecule and a biological molecule made up of CHO, right? And the chemical formula
Uh-huh.
of carbohydrates also you should write down, right?
Okay,
One
teacher.
carbon, two hydrogen and one oxygen, right? CH2O.
Okay.
Can you remember that one? So that thing also you have
Yeah.
to write down. And also you have to write down the different types of carbohydrates. That is monosaccharides, disaccharides, and polysaccharides, right?
Polysaccharide. Okay,
And
teacher.
the importance of carbohydrates you should write down. As an, what is the importance of carbohydrate? Energy source. energy source. You can write it down. Right?
Okay. Okay, teacher.
Right. So then just take your interval and come back on at 12.
Okay, teacher. Thank you.
Okay. Meow, මට ගන්න බැරි වුණා what only මේ ක්ලාස් එකනේ අද කෝල්ස් ආව කියන්නේ මෙයා තුනක් විතර ආවා හරිද එකකින් ආවා මේ සුප්‍රීම් ටීවී ප්‍රෝග්‍රෑම් බෑකෝ 80 මොන ෆීද 85යි කිව්වා ඔන්න ඉතින් ටීවී ප්‍රෝග්‍රෑම් එකකින් කතා කරනවා ඊට පස්සේ එක මිනිස්සෙක් මොනවාද චිකිත්සා මොනවද තියෙනවා ඒවට මොනවද ප්‍රොඩක්ට්ස් වගේ තියෙනවා හරි ඊට පස්සේ තව මිනිස්සයා මට බේරු වලින් කතා කරා මුස්ලිම් මිනිස්සු. එයා කතා කළා කියනවා "ඩි ડોක්ටර් බේරු වල දාන්න ක්ලිනික් එකක්." ටික අනේ වන්දන. ඒක හරි හොඳයි මේ මුස්ලිම් එකේ. මම ඔක්කොම බැලුවා ડોක්ටර්ගේ Facebook එක ඔක්කොම බැලුවා. හරි හොඳයි වීඩියෝස් එනවා. මේ බේන් වල දාන එක. ડોක්ටර්ස්ලා එනවලු ගොඩක්. මේ ඊට පස්සේ ඌ එහෙම කිව්වා. ඔන්න තව එකෙක් කතා කරා මේ මම මේ දැන් ක්ලාස් එකේ ඉන්නේ. එතෙන් එක්කෙනෙක් කතා කරා ඔන්න මේ මොරටුවේ ඉන්නේ කොයිද ගගා අහුවා. මම කිව්වා තාප්පිට කතා කරන්න කියලා. යන්න. ඔන්න ඔව් එයාට කතා කරන මම දැන් මේ ගිහිලි ඉවරයිලා. ඔන්න මොම් කට්ටිය තමයි කතා කරේ. ඔව් අන්න ඒකයි වැඩේ. දැන් අපි මෙහෙමනේ එකක් දාලා ඔව් අපි එහෙමනේ කරන්නේ. එකක් දාලා මාස දෙකකින් තුනකින් දාන්න ඒතර ඒගොල්ලන්ට බෑ. ඔව්. පිස්සුවෙක් වගේ නේද? ඒ තමයි මාත් මේ වීඩියෝස් හදලා දාන්න කියන මට වෙලාවක් නැහැ නේද? ආ එහෙමද? හ්ම්. ඔව්. ඔව් සමහර අය රෑ දකින්නවා. එහෙම නේද? ඈ ඔකට වීඩියෝස් ටිකක් හදලා දාන්න ඕනේ ගෑස්ට්‍රයිටිස් සෝරියාසිස්. හ්ම්. ඔව්. අර බූස්ට් කරන්න ඕනේ. හ්ම්. ඔව් ඒක තමයි හරි අමාරුයි මේ. ඔව් ඉතින් අමාරුයි එන්නේ නැහැ. ඒකනේ අර මේ හංසනිත් අර මේ දාලා දෙනකො පේෂන්ස්ලා නැල්ලු. ඌ දැන් ඒ කෙරෙන්ට් එක කියවන්න ඉන්දිය ඔප් එක්ක තියනවා. ආර වැඩ තියෙන්නේ. ඒක වහන්න මේ තෝ ඒක වහන්න බෑලු. නෑ අනිත් එක වහන්න ලැජ්ජයි මේ මේ කට්ටිය ගෙවල් ඉවලා මෙහෙන්නේ කට්ටියනේ මාළුදුරීමනේ. එකකුත් දාලා ඉල්ල වැහුවා. දැන් උඹත් වහන්න බෑ කියලා ඌ වෙනස වික්‍රී කියලා රෙට් එකේ. මාරු එතනදී ඉතින් බබු ගියා. ඒවනික අනිතිනේ අර රජයේ මේසෙ ඉන්න කපුනා නම් පොඩ්ඩක් අර මිනිස්සු මේ දෙන්න ගන්නව මත්. අනිත් එක මේ ගානත් අඩුයිනේ 1000යි මාසෙටම 1000යි. සති දෙකකට 500. එතකොට ටිකක් අර හොඳ උන් නැතත් එනවා. සල්ලි අඩුයි කියලා. බලමු ඉතින් එන්නේ. හරි හරි වෙනවා. බයි. Right. Just tell me what is a disaccharide? What is disaccharide? Yes, it is mainly made up of two monosaccharides. So how it is joined together? This is a single monosaccharide and another. That means the two monos are joined together by a bond, right? So this is a disaccharide. Now sucrose. Sucrose is table sugar. Is formed by molecule of glucose joining with the molecule of fructose. Fructose mean what? Monosaccharide. Glucose, monosaccharide. So these two monosaccharides are joined to form the sucrose, right? Sucrose is a disaccharide. Right. So the two monosaccharides join in a condensation reaction to form a disaccharide and water molecule is released. Now here when the two monosaccharides are going to join, just think this is glucose and this is fructose, right? These two are going to join together, releasing one water molecule, right? So when this bond is formed, one water molecule is released. And what is this reaction? Condensation reaction. So have you heard about these condensation reactions? Condensation reactions? Have you heard about the condensation reactions? When these two are going to join, one water molecule is released. So that is a condensation reaction, right? So what is the bond which is formed in between these two? What is the bond formed? That is glycosidic bond, right? So in a condensation reaction, one water molecule is going to be released and the bond formed is called glycosidic bond. What is it? What is the bond? Glycosidic bond, right? So we use numbers to show which carbon atoms are involved in this bond. If carbon 1 on one monosaccharide joins to carbon 4 on another monosaccharide, we called it 1,4 glycosidic bond. Right? That means what? In one monosaccharide, if the carbon 1 of one monosaccharide is going to join the carbon number 4 of another monosaccharide, then the bond which is formed is called the 1,4 glycosidic bond. Understood? Understood? No.
Yes, teacher.
So there is two molecules like this, right? Now just think this is one monosaccharide, right? And this is another monosaccharide. Now in the disaccharides, bond is formed like this, which called the glycosidic bond, right? And the reaction is condensation, finally removing one water molecule out, right? So in this monosaccharide, there is carbon like this. In here also, in this monosaccharide also like this. There is carbon like this. So the carbon number 1 is joining with the carbon number 4 of the second monosaccharide. Understood? Carbon number 1 of first monosaccharide is going to join with carbon number 4 of the second monosaccharide. Then what is the bond? 1,4 glycosidic bond. Why is 1,4 glycosidic bond? Because carbon number 1 of this monosaccharide is going to join the carbon number 4 of the other monosaccharide. Right? So therefore we call the bond as 1,4. Understood, Savindi?
Yes, teacher.
Right. So we use numbers to show which carbon atoms are involved in the bond. If carbon 1 on one monosaccharide is joining the carbon number 4, then we called it 1 to 4. Right? 1 and 4. So if the bond is between carbon 1 and carbon 6, 1,6 glycosidic bond, right? So this is the reaction that they have given, right? Here. Just see, this is an alpha glucose, right? So you know what is alpha glucose. What is alpha glucose? Can you remember what is alpha glucose? Alpha glucose are the monomers of, sorry, isomers of glucose, right? Isomers of glucose. Now just see here, OH group on the, huh? Yes, below the plane. Below the plane. So here, just like that, here they have given one alpha glucose. OH is below the group, below the line, right? Below the plane. Here also an alpha glucose. Now here they are showing condensation reactions. How this condensation reaction is going to happen? By removal of water molecule. How the water molecule is going to remove? OH from this one and H from the other monocyte. Right? So then one water molecule is going to remove. Right? This is the condensation reaction. And here OH and H is not there, so only the O bond. So what is the bond which is formed here? What is the bond which is formed here? 1 and 4. 1 and 4. Why? Because carbon number 1 and carbon number 4, right, is going to form the bond. So it is 1,4-glycosidic bond. Understood? Right. So then they have given 3 different types of disaccharides, right? So what are the disaccharides they have given? Sucrose, lactose, maltose. How the sucrose is formed? How sucrose is formed? Glucose plus fructose. You have to remember these things, right? This is very important. Sucrose, because in MCQs they can ask. Sucrose is formed from glucose and fructose. So it is a disaccharide. Lactose, glucose plus galactose. What about maltose? Glucose plus glucose. Where can you find sucrose? Hmm? Huh? Fruits. Yes. So that is fruit, right? Normally the table sugar you can call it like sucrose. So what about lactose? Hmm? Milk. In milk you can find lactose. Maltose? Seeds like barley, right? Okay, they have given questions. What are carbohydrates? You did this one, right? Describe how a glycosidic bond is formed between two monosaccharides to form a disaccharides. Can you do that? Describe how a glycosidic bond is formed between two monosaccharides to form a disaccharide. Just write down the answer. Finish writing? Finish? Yes, Samiti, what is your answer?
When two monosaccharides are joined by a condensation reaction removing one molecule of water.
Yes, condensation reaction, right?
Yeah. And creating a covalent bond between the sugar units. Am I correct?
When two molecules of monosaccharides are joined together, one molecule of water is going to remove. So that is known as the bond formed is known as a glycosidic bond, right? Now here, in this type of question, it is better to include the diagram like this, right? First, you can draw this one. Try to draw it, right? So after going home, you just practice drawing this one because then you can see how the OH from one molecule of monosaccharide is joining with H, right? That means it is going to remove one water molecule. So this is the condensation molecule, sorry, condensation reaction. And by O, that means oxygen, this is going to bond, right? And you can show this type of bond also here, 1,4. So just try to practice drawing this diagram, right? Right, then the practical skills. Now here, these practicals you have to study. If you came to this orientation, you know that your units 1 and 2, right? The practicals that you're doing in unit 1 and 2, you have to uh, write an examination in unit 3, right? Unit 3 comes as all the practicals. Now here, the first one that they have given here, I think I have included only this practical for this presentation only, right? So all the other practicals we are going to do separately. Then you can understand, right? This is just, we'll see what is this practical, right? Here we are going to test the sugars, right? So have you heard about this Benedict solutions? Benedict, have you heard? Right. So why are you using the Benedict reagent? Benedict solution? Have you done this experiment in your labs, school labs? Yes. Yes. So then you can't remember what is the Benedict solution and why you're using this Benedict solutions? What is the color of the Benedict reagent? Hmm? Brown. Huh? Huh? Brown. Brown. So in your school, huh? Blue color. Here they are telling, right? The Benedict solution is a chemical reagent for testing reducing sugar. So what is the reducing sugar? Have you heard about the reducing sugars? Okay, now you have heard about the Benedict reagent in your school. That means you have done the experiment. Experiment also you have done. Huh? Done. But you don't remember now. You have completed your O levels recently, and you can't remember why you are using the Benedict reagent. Reducing sugars. Hmm? What is a reducing sugar? Yes, Savindi, what is a reducing sugar? Reducing sugar means a substance which is going to donate electrons, right? Reduce, reduce by donating electrons, right? Now here, Benedict solution is bright blue. Why it is bright blue? Because it is going to contain the copper ions, right? It is going to contain the copper 2 plus, 2 ions. So then it is going to donate the electrons. So we call that as a reducing. Now here you are going to use this Benedict reagent in order to find out the reducing sugars, right? Now some of the sugars react readily with the solution when heated gently and reduce the copper ions to copper 1. Copper 2 to copper 1, they are going to reduce it by donating electrons. So a precipitate is formed and a color change is from blue to orange. Now have you seen this color change? Have you seen the color change? So blue directly is going to change the orange. So then, hmm? Yes, this is the color change. I didn't put it. This is the color change, right? Blue, green, yellow, orange, and red. So this red is brick red precipitate, right? It is going to form a precipitate. You know what is a precipitate? Yes. So this is the color change, right? It, now here there is a small note about the practical, right? But you have to do this practical again. So don't worry if this is not understanding, right? So there is another part that you have to, this is the practical one, right? This is the first one that you have to do. But I have included here also. This is after completing this one, you have to, after completing the chapter, you have to do the practicals, right? So I'm going to do all these practicals separately. So then you can understand, right? So there is separate practical list, and for A2 there is separate practical list, right? So sugars that react in this way are known as the reducing sugar. So the sugars which is going to change the color from blue to brick red precipitate, we call it as what? Reducing sugars. So all of the monosaccharides and some of the disaccharides are reducing sugars. So by after doing this experiment, you can find out all the monosaccharides, they are reducing sugars. And some of the disaccharides, not all, except sucrose, right? It is not going to change the color. Now, the sugars which is not going to react with this Benedict reagent is known as what? Non-reducing, right? So can you just give me an example for reducing sugar? Yes, what else? Yes, then non-reducing sugar? Sucrose, right. Now here you can heat a non-reducing sugar like sucrose with a few drops of hydrochloric acid, that means HCl, Allow it to cool and then neutralize the solution with sodium hydrogen carbonate to hydrolyze the glycosidic bonds. This produces the monosaccharide units of the sugar, which will give a positive Benedict test. Now, what they are telling here? Now, you know that sucrose is non-reducing sugar, right? You can't see the color change from blue to brick red precipitate. But if you're going to add this hydrochloric acid, HCl, and also sodium hydrogen carbonate, what happens? It is going to, yes, why it is going to change the color? Because what happens to this glycosidic bond? It is going to hydrolyze. What is hydrolyzation? Condensation and hydrolyzation, right? Now here's the C. This is the one monosaccharide, and this is another monosaccharide, right? So in order to form the bond between this glycosidic bond, what is the reaction? It is condensation, right? Condensation means it is going to remove one water molecule, right? So in this reaction, it is hydrolyzed. Hydrolyzed means what? Hydrolyzed means? Hydrolyzed means opposite of condensation reaction, right? Opposite. Now, condensation means removing of water. What is hydrolyzation? Addition of water. Addition of water, right? So, when you are adding hydrogen, this HCl and hydrogen carbonate, what happens? The glycosidic bond is going to break, right? Break. How it is going to break? With the addition of water, right? So when it is going to break, what happens? Monosaccharides are going to form. Now sucrose, how it is made up of? Sucrose. Sucrose is a disaccharide. What are the monosaccharides there? Huh? Fructose and glucose, right? So those are monosaccharides. So when it is going to break the bonds, so these monosaccharides are separating. And then you are going to perform this Benedict test, and then you can get the positive result. Understood? Understood this one, Savindi?
Yes,
Got
teacher.
it? Right.
Yes.
So here, always remember condensation means removal of water, right? To form the bond, to form the glycosidic bond, there should be removal of water molecule. But in hydrolysis, what happens? Addition of water, right? Addition of water. Addition of water and breaking the bond, right? Glycosidic bond can be break, right? And then polysaccharides. So what do you know about polysaccharides? Yes? Hmm? What? Yes, more than 2 monosaccharides. So it is a polymer, right? The most complex carbohydrates are the polysaccharides. Can you give me 1 or 2 examples of polysaccharides? Yes, what else? Cellulose. So these are made up of many monosaccharide units joined by condensation reactions to form the glycosidic bond. Now here, as it is a chain of reactions, chain of monosaccharides, right? Like this. Several monosaccharides are going to join by glycosidic bond. So what is the reaction that is happening here? Condensation, right? Condensation reactions mean at each of the steps condensation. And polysaccharides do not have sweet taste. It is not sweet. Molecules with between 3 and 10 sugar units are called the oligosaccharides. while molecules containing 11 or more monosaccharides are known as the true polysaccharides. And there is two names also, oligosaccharides and polysaccharides. Oligosaccharides mean the units are 3 to 10. If the units are more than 10, sorry, more than 11, we call it polysaccharide. True polysaccharide, right? Now, the glycosidic bonds in the polysaccharides can be broken to release monosaccharide units for the cellular respiration. Right? How it is going to break? How these bonds are going to break? How the glycosidic bonds are going to break? Hmm? Monosaccharides units are going to join with glycosidic bonds by condensation reactions. Now, these in polysaccharides, monosaccharides can be separated by breaking the glycosidic bonds by using which reactions? Hydrolysis, right? Hydrolysis. Right. The glycosidic bond between two glucose unit is split by a process known as hydrolysis. The hydrolysis reaction is opposite of the condensation reaction. What is added to the bond, right? Give me two differences between these condensation and hydrolysis reactions. Hmm. Yes, so that thing you have to understand, right? So starch and the glycogen are gradually broken down into short and short chains, and eventually single sugars are left. Glycogen, starch, everything is polysaccharide. Disaccharides break down to form two monosaccharides. Hydrolysis takes place during digestion in the gut, also in the muscles and the liver cells. When the carbohydrate stores are broken down to release sugars for use in the cellular respiration. So normally for the cellular respiration, this breaking down happens, right? Using hydrolysis reactions. Why? Because they are going to provide what? Monosaccharides. Monosaccharides mean for the cellular respiration, it needs what? Glucose. Glucose is going to provide energy, right? So that's why. That's why these hydrolysis reactions take place. Carbohydrates and the energy. Every chemical reaction taking place in cell needs energy. Energy is supplied by substance called adenosine triphosphate. Have you heard about ATP? Right.
Yes,
So this, yes. Yes, Avinti, did you tell anything?
no teacher.
Right. So this ATP comes from the breakdown of the monosaccharide glucose using oxygen in the process of cellular respiration. So can you remember the equation of cellular respiration? Hmm? What is the equation for photosynthesis? Sorry, the formula or the reaction? Photosynthesis, I'm asking. Reaction? Yes. You did single amino. Okay. So then that means O levels also you did single amino. So it is okay to catch the words. No problem, right? Okay. So if you find any difficulty, just ask me, right? Anything. Now I am asking, what is the formula for this photosynthesis? Six. Yes. So here they have given the cellular respiration, right? So what is the cellular respiration? C6H12O6 means glucose, right? With the presence of oxygen. So then it will give carbon dioxide as a byproduct, right? And water, energy. Energy mean ATP. This is the cellular respiration. So for the cellular respiration, mainly glucose is needed, right? So that's why hydrolysis reactions taking place, right? Starch is breaking down to produce this glucose. So glucose is involved in the cellular respiration, right? So this is the reaction. Just remember the chemical formula. This respiration and all you have to study it again in A2 syllabus, right? This process occurs in the mitochondria of the cells and is essential for the producing energy that the cell can use for various functions. Now, cellular respiration take place in respiration take place in which organelle? Organelles. Now, in animal cells, what are the organelles that you can find in animal cells? I think you have studied, right? No? Animal cells, nucleus, mitochondria, right? Golgi bodies, ER, endoplasmic reticulum. Have you heard ribosomes? Right. So they are in these cells, you can find this mitochondria, right? In mitochondria also there are different. Now, if you consider about this mitochondria, you can draw it like this, right? But there is different parts inside this mitochondria, right? There are striata and then matrix and all like that. So this respiration is going to take place in different parts of this mitochondria. But generally we are saying the respiration takes place in mitochondria, right? Any glucose in the food you eat can be absorbed and used directly in your cells. Other monosaccharides and disaccharides, for example, fructose, maltose, sucrose, they are also easily absorbed in the body and rapidly converted to glucose. So why it is going to convert to glucose? By hydrolysis. To produce energy, right? So foods containing monosaccharides and disaccharides are good source of relatively instant energy. However, this cannot be used to store energy because they are chemically active and they are very soluble in water. So they affect the water balance of the cell. Now here, if you consider about this glucose and all, they are not going to store, right? You can't store it. Why? Because they are so much chemically active. So that's why they are going to store in like starch, glycogen, like that, right? Then in need only, they are going to break down, right? To produce energy. Right. Then structure of the polysaccharides. They have given some polysaccharides. Right. So I will give you now one question. Just try to answer this one. Explain why. Explain why sugars such as glucose and sucrose, glucose and sucrose are useful for immediate energy. Are useful for immediate energy, but are not suitable as long-term energy stores. Long-term energy stores. Finish writing. Please. Right now, here they are asking why this glucose is not suitable for long-term energy, right? Energy source. Now we just see this glucose and sucrose. What is glucose? Glucose is monohydrate. Sucrose. Yes. So just glucose here we are talking about glucose. Right? So these are very much, first thing is very much soluble. Right? This is very much soluble. You know that? This is very much soluble, right? That is first thing. When it is soluble, more soluble in water, what happens? What happens to the osmotic pressure? Have you heard about this one? Osmotic pressure? No, no idea. Right. Now, when it is dissolved, what happens? The soluble concentration will be high. Right? So, as it is more soluble, it is going to soluble, that means efficient. Right? So, their osmotic pressure will be high. Why the osmotic pressure is high? Because solute level, solute concentration of this is high. What about the water content there? Solute is more. What about the water content? Solute content is more. What about the water content there? Water? Low. Water content is low. Why? The more solutes is dissolved. Water molecules is less now. Right here. So therefore the water pressure is also water potential. Water potential with pressure is less. Right? So here when you are considering about this glucose and sucrose, as it is more soluble, it can create an osmotic pressure. Right? That means as it is going to dissolve more. Right? So then what happens if there is an osmotic pressure in this living medium? There is an imbalance of water. Right? There is a water imbalance. Water imbalance. So that's why there is one reason why this is not suitable. Right? For long term. Why this is not suitable for long term? That is one thing. Soluble and it can increase the osmotic pressure. Right? That means it is osmotically active. Right? Osmotically active. Soluble, osmotically active. Understood? This is first reason. And the other one is this is more reactive. Right? This is more reactive. That means chemically reactive. Chemically reactive. Right? Than these polysaccharides. Chemical reactive. If it is chemically reactive, what is the disadvantage of that? Why you can't keep it for this long-term use if it is chemically reactive? Chemically reactive means it is going to become more unstable. Right? It is going to interfere with other functions also in the living body. That's why you should, we are telling that this is not suitable for long-term use. Right? Glucose and sucrose. Understood? One thing is it is very much soluble. So it is going to have the osmotical effect or osmotically active. Right? and then other one is chemically reactive. So these are the main reasons why it is not suitable. Now, if this can create this osmotic imbalance, what happens mainly? Now, just I'm telling you this one, right? Now, just think this cell, right? This cell contains more solutes, right? Now, what happens to the osmotic pressure in this cell? If it is going to contain more solutes, what happens to the osmotic pressure? It is osmotic pressure more solutes, then osmotic pressure is high, right? Why? The pressure which is exerted by the solutes, it is high. So then, what happens to the water potential then inside the cell? It is low. So then what happens? Normally, in I think in the other lesson you have to learn about this one, cell membranes and all. When this normally water is going to move from high concentration to low concentration of water, right? High concentration to low concentration of water. Now just tell me here, what is the water concentration inside the cell? Inside the cell, water concentration is low. Low, right? So, as the water concentration is low inside the cell, what is going to come from out to inward? Right? Then what happens to the cell? It will, what happens? Now, cell is like this. What is coming from outside? What happens? It will, hmm? I'm showing the animation, still you can't tell. Cell is like this. What is coming inside and it will be like this. What happens? Swelling, right? Swelling. Because what is coming from outward, the cell is going to swell. Swelling and swelling more. Then what happens? Finally, cell will burst. Right? Finally, the cell will burst. So that is the reason, right? So that's why this osmotic pressure is important. So in this answer, you can write about this glucose and disaccharides are more soluble. So they are going to create the osmotic problems, right? As it is soluble, osmotic problems in the cells. So as it is osmotically active, it is going to increase the osmotic pressure. Did you write the answer? Do I want to repeat it or do I want to tell it for you to write? Right, just write down this one. It is too soluble. Glucose and this sucrose, right? It's too soluble. They are highly soluble. So they would cause osmotic problems in the cells. So they cause osmotic problems in the cells. So you can write within brackets, draw in water and burst the cells. Draw in water and burst the cells. They increase the osmotic pressure of the cells. They increase the osmotic pressure of the cells. They increasing the osmotic pressure of the cells, disrupting water balance. Disrupting water balance. They are chemically reactive and unstable. Chemically reactive and unstable. So interfere with other cell functions. unstable, and may interfere with other cell functions. So for long-term energy storage, in animals, glycogen, and in plants as starch is used, right? Then right. So do you have any questions to ask from this part? No, understood, right? So then we'll go for this polysaccharides. The structure of the polysaccharides make them, did we discuss this part? No, no. Right. Structure of the polysaccharides make them ideal as energy storage molecules within the cell. Now here, as we know that these monosaccharides and disaccharides are not so much important for energy storage. Now you know what is the answer, right? Why we are calling that it is not important for the energy storage. But these polysaccharides are important for energy storage. Now can you guess why these polysaccharides is important in energy storage? Hmm? Less soluble, that is correct. What else? Yes, these are chemically inactive. What else? As it is not going to soluble in water, what happens to the osmotic pressure? Low. It's not going to cause any osmotic problems, right? So that is the reason. That's why it is going to store as polysaccharides. Right. Savindi, did you understand this one?
Yes, teacher.
Right. So we'll start the first one, that the polysaccharide starch. Right? Do you have any question? Sorry, any idea about this starch? Hmm?
Made of glucose molecule,
Yes, that is polysaccharide, right? So what else?
energy storage, carbohydrates in plant,
Yes, in plants, it is an energy storage. What else? As starch is an polysaccharide, you can tell all the features of polysaccharides here, right? It's not going to dissolve in water. It is insoluble. It is compact. What is this compact mean? No idea. Right, we'll see what is this compact and all later on, right? The sugar produced by the photosynthesis are rapidly converted into starch, right? From the photosynthesis, sugar is produced and that is converted into starch. Why it is converted into starch rapidly? From photosynthesis, what is producing? Glucose. Why it is converted into starch rapidly? To store in plants. Because you know what is the reason now, right? It is not going to store as glucose. Right. So that's why it is going to store as starch, which is insoluble and compact, but can be broken down rapidly to release glucose when it is needed. When energy is needed, it is going to break down with, what is the reaction? Hmm? With what? With water. What is the reaction? Hydrolysis. Right. So starch consists of long chains of alpha glucose. Starch is having alpha glucose. You know what is alpha glucose now? Right. But if you look at it more closely, you will see that it is a mixture of two compounds. So in starch, you can find two compounds. One is amylose, other one is amylopectin. What is this amylose? Amylose mean, now in question papers, they have asked the differences, I guess, between these two. Right, amylose and amylopectin. I have seen. Right, amylose mean it is an unbranched polymer. So all these things are polymers, right? The starch mean polymer. It is an unbranched polymer between how many number of glucose units here? 200 to 5000 glucose unit. So as the chain lengthens, the molecule spirals. Spirals mean molecule is going to spiral, right? Which makes it more compact for the storage. So now the linear structures. Linear structure is like this, right? This is the linear structure. This is the linear structure. Spiral structure is like this. So which is from these two structures, which is the most important one for storage? This is very much lengthy, right? Linear. This is lengthy. That's why it is going to spiral, make spirals. So which one is beneficial in storage? Huh? Second one, spiral one, right? It is important because it is going to become compacted, right? It's going to compact it because of the structure. So it is important for the storage. And amylopectin is branched polymer, right? Amylopectin means branched. So this is unbranched, linear, and due to its length, it is going to spiral, right? Amylopectin means branched. The branching chains have many terminal glucose units that can be broken off rapidly when energy is needed. Right? Now these branched polymers having terminal glucose molecules. So when energy is needed, it is going to break efficiently. Right? So that is the difference between these two. Amylose and amylopectin are both long chains of alpha glucose units. So you have to know that both of these are alpha glucose. But what is the difference between these two, amylose and amylopectin? Yes, amylopectin is branch one, right? Now, if you consider about the energy supply of these two, which is more important in energy supply? Hmm? Amylopectin. Why it is important more? What? Huh? Small? Yes, because it is having terminal glucose, right? When needed, it is going to supply energy, right? That is the reason, right? Amylose and amylopectin are both long chains of alpha glucose units. So why are the molecules so different? It all depends on the carbon atoms involved in the glycosidic bonds. Now in bonding, these carbons are different, right? So their structures are different. So that is the reason why these two are having different structures. But their uh, structures and their use is also different, right? So here amylose has only 1,4 glycosidic bonds. Can you explain that one? Amylose is having only 1,4, but amylopectin, it can have 1,4 and 1,6 also. What is the reason? Amylose is having only 1,4, right? Amylopectin 1,4 and few 1,6 also. Why is that? Because of the structure. No, now this amylose is only a linear one. So that's why it is having only 1,4. But this one is, it is branched. So it can have the bonds with other carbon molecules also. So that's why it is having 1,6. Understood?
Yes,
So starch has a combination of straight chain amylose and branch chain amylopectin molecules. So in starch, it is having two compounds. One is amylose and the other one is amylopectin. The combination explains why carbohydrate foods like rice and pasta are so good for you when you are doing sports or hard physical work. Right? The amylopectin releases glucose for cellular respiration rapidly when needed. Amylose releases glucose more slowly over time, keeping you going longer. That is the difference between these two. Can you understand that sentence? What is the difference between amylose and amylopectin in releasing energy? Amylopectin releases energy rapidly. Why? Because of the structure, right? The terminal glucose you can find. But amylose, it is going to release glucose slowly because of the linear structure, spiral structures, right? So this is the picture of amylose and amylopectin. Here, amylose. Amylopectin branched, right? Here, this this is one branch. In this chain, you can find 1,4 glycosidic bonds, and here in this branch, you can find 1,6, right? Okay. So now you have to tabulate the differences between these two amylose and amylopectin, right? Differences between amylose and amylopectin. Just draw a table like this. Differentiate the differentiate amylose and amylopectin, right? Like this table. One is amylose, other one is amylopectin. Right. Then right, one, two, Finish. Right. When you're writing the differences, you can write about the structure mainly, right? So amylosis branched, right? Amylopectin is sorry, amylosis unbranched. Amylosis unbranched, amylopectin is branched.
Branch.
Yes. So then, what about the shape? Okay, just tell me the bonds that is there in amylose. Yes. So then, what about amylopectin? Right. Shape. Hmm. Huh? Amylopectin is branched, right? It is branched. What about the solubility? Amylopectin is what? Amylosis. Amylopectin is more soluble and amylose is less soluble. Is that? Yes. And then what about the energy release of these two? Energy release, it is important, right? So you can add up the points. If you didn't write this, you can add. What about the energy release? Hmm? Yes. Yes. Right. Okay. So those are the differences between these two. So do you have any questions to ask? Amylose is linear. Amylopectin is branched polymer, right? Branch polymer. I guess they didn't have. Here they have given one diagram like this, right? Here, amylose, linear, amylopectin, branched, right? Can you see that? That is the shape or structure. Right. Then And so we will go to glycogen also. What is glycogen? Polysaccharide, right? So animal starch. It is animal starch because it is only carbohydrate. It is the only carbohydrate energy store found in animals. In animals, energy can be stored in the form of glycogen. So it is also important in important storage carbohydrate in fungi. Fungi also glycogen is important for the energy store, right? Glycogen is very similar to the amylopectin molecules in the starch. So then give me some example, give me some characteristic features of this glycogen. If it is very similar to the amylopectin molecules. Hmm? Amylopectin. Hmm? Yes. Like starch, it is very compact. But the glycogen molecule has more 1 to 6 glycosidic bonds than the starch molecule, giving it many side branches. Now in the glycogen molecule, see the difference, right? This is amylopectin, this is glycogen, but can you see how many branches that is there in glycogen? It is more, right? Compared to the amylopectin, glycogen is having more. What? Compared to the amylopectin, glycogen is having branches, more branches, right? So more branches, so it can produce more glycosidic bonds, 1 to 6 glycosidic bonds than starch. This means that glycogen can be broken down very rapidly. Why it is going to break down very rapidly? Because it is having more side branches, right? The terminal branches. So this makes it an ideal source of glucose for animals which may require rapid release of energy at certain times of high activity levels. Now, normally animals, they are having high activities, right? So in that case, this glycogen can provide more energy when needed. So that's why it is important, the side branches. And they have given one diagram here, right? Glycogen. This is the branch, right? So here you can understand what is the difference between these two. Right, do you know how to test for starch? Have you heard about this iodine solution? Right? So what is the color of iodine? Hmm? What is the color change then? Hmm? Huh? Yes, blue black, right? Bluish black. So here, if you want to identify starch containing materials in the laboratories, you are using this iodine solution. What is the color of iodine solution? Mainly it is blue, reddish brown. It is reddish brown, right? Reddish brown mean reddish brown mean reddish brown mean red and brown both, right? Reddish brown. So if you are going to add this iodine solution to any material which is containing a starch, what is the color change which is taking place? Reddish brown to bluish black or blue black, right? So this is also one of the co-practical that they have given. Right, now you have to do these co-practicals. First one, the time. Right, before moving into this one, can we do a one question? Mhm. Yes, write down this one. Now I have done some of the checkpoint questions here in the student book, right? I have given also one or three questions to do at home. So I have to do it. So this is also one of the checkpoint questions they have given in the book. Just write down. Explain how the structure of carbohydrates, explain how the structure of carbohydrates is related to their function. How the structure of carbohydrates is related to their function as storage molecules providing the fuel for cellular respiration, providing the fuel for cellular respiration in animals and plants. In animals and plants. Right. Explain how the structure of carbohydrates. Explain how the structure of carbohydrates is related to their function as storage molecules. Related to their function as storage molecules, providing the fuel for cellular respiration, providing the fuel for cellular respiration in animals and plants. Hello. Huh? Huh? දෙකටනේ. අරයා මොකද දැන්? Finish writing. Finish. Finish. Right. Savindi. Finish.
Teacher.
What?
Yes, teacher.
Right. Just tell me your answer.
The structure of carbohydrates makes them ideal energy storage molecules. They are made of glucose, the fuel for respiration. Their glycosidic bonds store energy and can be easily broken to release glucose. Uh,
That's all.
Yes, teacher, I have to write right.
Okay. Now, this question is asked how the structure of carbohydrate is related to the function as storage molecule, right? Carbohydrate and storage molecule. So, storage molecules mainly we have to talk about what? As the storage molecules, we have to talk about what? Huh? Glycogen and starch. Right now, if you consider about these two, these are mainly made up of, right? You can tell alpha glucose, so just glucose monomers. Then they are having the compact structure. If you consider about the compact structure, we have talked about this amylose, right? And then we talk about the aminopectin, right? So we talk this one as a linear spiral. So what about this compactness? What about the compactness here? It is going to form a helix. So you can compact it for the storage. So this is an as when you're talking about this amylose, it can create this compactness using the structure, spiral structure, right? So when this linear structure is going to link like this, it is going to spiral, make the spirals like this, right? So it is going to compact. So that is one thing. And then glycogen, it is having highly branched structure. So they are going to pack densely, right? So then whenever in any need, it is going to give out the energy out, right? And the other one is, it is insoluble. Insoluble nature of the storage carbohydrates. That means glycogen and the starch. It is insoluble, right? Right? Is it starch and glycogen? Insoluble or soluble? Yes, comparatively it's less soluble. So if it is going to less soluble, then what happens to the osmotic pressure? low and it is not going to create any osmotic affection, right? So therefore it is not going to balance, not going to affect for the water balance, right? So that is also one thing in this storage. And the other one is, yes, mainly you have to write about this glucose molecule. It is the glucose molecules are going to, or you can write this made up of glucose monomers, right? And compact structures. And they are going to insoluble in water. And branch chains. Understood the question answer? Right. No, I will read out the answer for you, right? Then you can just write it down. Now the question is says like, can you understand the question first? Tell me. Can you understand the question? Right. The question is asking how these structures of the carbohydrates is related to their function as storage molecules, right? Storage molecules, when you're talking, mainly you have to talk about this starch and glycogen. Starch in plants and glycogen in animals, right? So you can tell the first point as they are made up of glucose, right? So these two molecules are polysaccharides and they are going to made up of glucose monomers, alpha glucose mainly, right? And then the compact structure, second point, the compact structure. Compact structure, when you are writing, you can write about the amylose, right? It's going to coil, so it can make compact. And glycogen is highly branched, so it is going to densely packed, right? So what is the importance of this compactness? More energy is going to be stored in less space, right? Understood. Now we are talking about the compactness here. This linear structure, this linear structure is going to become spirals like this. So they can compact, right? So in a less space, more energy can be stored, right? Understood? In less space, more, and then insoluble in water. This starch and glycogen, they are insoluble in water. They are insoluble in water. What is that? Savinthi, can you hear me? Savinthi, can you hear me?
Yes, teacher.
All right. So So insoluble in water, right? So if it is going to, if it is not soluble in water, it is not going to affect the osmotic balance, right? Then they are having branch structures. So this branch structure, it is helping for the energy release, right? Amylopectin and the glycogen, they are having branch chains at ends, so they can rapidly release glucose when energy is needed. Rapidly release glucose when energy is needed. So this is the thing, right? So starch and glycogen is mainly adapted to act as storage molecules. Understood? Now, any questions to ask? Anything to clarify? No? Yes? No, right? Okay, then co-practical one, right? I have discussed this one a bit with you. That is about the Benedict solution, Benedict reagent. Right. The first practical is like this. Uses semi-quantitative method with Benedict's reagent to estimate the concentration of reducing sugars, and with iodine solution to estimate the concentration of starch using color standards. Right? What is this semi-quantitative mean? Can you understand what is the topic here? Benedict's to test? Which one? Reducing sugars. An iodine solution to test? Starch. Now, what is this semi-quantitative mean? What is quantitative? What is quantitative mean? Quantity. Quantitative mean? Huh? Hmm? Pramana. Right. Pramane, right? Quantity.
Estimate
Semi-quantitative.
value,
What?
estimated value.
Yes. Estimated. That means approximately we are going to measure. Right? Approximately we are going to measure. So that is the semi-quantitative. Right now here, we don't have the labs to perform the experiment, but you also have this examination paper to write out, right? So just see this one, how to do the experiment. I think you all have the experience in the laboratories with the beakers, Bunsen flames and all, right? You have seen it and you have touched it and you have do the experiments, right? That also. Okay, so all the sugars can be classified as being reducing or non-reducing. So if you consider about the sugars, carbohydrates, you can classify it as reducing and non-reducing. So you know what are the examples for reducing and what are the examples for non-reducing, right? And you know how this non-reducing sugar can be converted into a reducing sugar also. Can you remember that one? How we are going to convert? Hydroxyl. Hmm? Hydrochlor, HCl, right? Okay, so a test with Benedict reagent can not only reveal if it is a reducing sugar or not, but also how strong of reducing sugar. It is not going to reveal only the Only that reducing sugar is strong. Sorry, reducing sugar or not. But how strong the reducing sugar is, right? This test can show. Right. So first you have to collect all these equipments for the experiments. What are the equipments that you need? Benedict reagent. That is the first thing, right? Benedict reagent. What is the color? Test tubes. Water bath set to 90 degrees Celsius. And then you have to get pipettes. Have you seen these pipettes? Pipette? No? Test tube rack, a stopwatch, right? So all these equipments you have to take. Have seen the pipette glass one? No? Right. So method. Now, if I have this, I have some of these videos. I'll put it to the group, right? So if I find it, so you can just watch it and see the method. Pipette 5 cubic centimeter of the solution being tested into the test tube, followed by 2 cubic centimeter of Benedict reagent. Right? Now, first you have to take the solution that you are going to test to a test tube. Have you seen the test tubes? Right. So, what is the amount? 5 cubic centimeters. 5 cubic centimeters mean what? In milliliters? Five cubic centimeters in millimeters, milliliters. One cubic centimeters is equal to how many milliliters? Right. So followed by 2 cubic centimeter of Benedict reagent. Right now, first you have to take to the test tube 5 cubic millimeter of the solution that you're taking and 2 cubic centimeter of the Benedict reagent. Today, you go home and find out the relationship between cubic centimeters and milliliters, right? Okay. So place the test tube in a test tube rack and then into the water bath and leave it for exactly 2 minutes. What you have to do? You have to keep this test tube in the test tube rack and then you have to keep it in the water bath and you have to leave it for 2 minutes. Keep it there for 2 minutes. Then remove the test tube and observe its color. Then you can observe the color change. If it is reducing sugar, what happens? Now here it says, this is semi-quantitative. Why? Observing the color change on the scale from blue to red, we can estimate the concentration of the reducing sugar, right? So that is the reason why they are saying it as a semi-quantitative. Now what is the color change? Blue color to brick red precipitation. So that is very important, right? You don't write it like a red color only. It is a precipitate. It can form a precipitate. So you have to write from blue to brick red precipitate. So blue color is a negative result, right? If that blue color is not changing, it is negative. That means it has to be a non-reducing, right? So blue color is a negative result since this color of the Benedict reagent, while color closer to the brick red gives a positive result, right? With an increasing concentration of the reducing sugar. So you can find out how the concentration, what is the concentration of the reducing sugar also. How you're going to find out that? By observing the color change, right? By observing the color change. How bright the color is, right? Now, sometimes the color will stop from the orange, right? So likewise, you can find out what is the strongness of this reducing sugar. Understood? Right. So here, this is the color change that you can get from blue to brick red. Blue is negative result, right? This is negative and brick red precipitate is positive. Right, the method. Right, this is the starch. Here, pipette 2 cubic centimeter of the test solution to a test tube and add 2 drops of potassium iodide solution. What is the color of potassium iodide? Hmm? What is the color of potassium iodide iodine solution? Yes, they are asking. Huh? Brown. Which brown? Reddish brown. Right? So a color change from brown to bluish black. It indicates the presence of starch. So this is the starch experiment, right? So the color change will be from reddish brown to bluish black. Use of color standards. The concentration of both the starch and the reducing sugars can be estimated using color standard, which is when the Benedict test and iodine test for starch are carried out on known concentration of starch and the reducing sugars. Right. Now here you can find out what is the concentration of the reducing sugars that is there or the starch, right? The colors produced are known as color standards, to which the results of testing unknown solutions can be compared to estimate the concentration. Look at estimate the concentrations by doing this experiment. And also you have to know what are the risks that is associated, right? Now, when you are using this experiment or when you are performing this experiment, you can have liquids and you can have hot water bath and you have glasswares. So what are the risks associated with using of this, right? When you are using liquids, what is the risk? What are the liquids that you're using here in this experiment? Hmm? Water, right? What else? Liquid mean there. What are the liquids that you're using in the experiment? Benedict reagent, right? What else? Dear lamai. One of the dear which we are using in the experiment. Dear Benedict එකයි අනිත් එක මොකද්ද? Iodine solution එක. Those are liquids, right? So what is the risk associated with these liquids? Spillage. Spillage spillage that could cause surfaces to be slippery, leading to an accident. Spillage mean හැලෙනවා, right? It can cause accidents because it makes the surfaces slippery. That is one risk associated with using of these liquids, right? So what is the precaution that you can take for this? What is the precaution that you are taking for this? Huh? Yes, wiping. Wiping here, wiping any liquid spillage as soon as they occur, right? And then you can put the lids to the bottles. And then after using the bottles, you can keep it away and keep away from the edge of the desk. So all these things are precautions that you can take, right? For this risk. And the other one is hot water bath. What is the risk? Yes, scalding. Then precaution. Take care in removing and replacing the water bath lid and have a first aid kit nearby. Remove test tubes from the water bath with tongs. Keep away from the edge of the desk. Now, when you are using this hot water bath, you have to be careful, right? Because it is 90 degrees Celsius, right? Can you remember the temperature? 90 degrees Celsius, right? So it can cause scars. So here when you are using these test tubes in the water bath, you have to use tongs. What do you mean by tongs? Test tubes you are not now. Just think this is like water bath, right? This is the water bath, cold water bath, 90 degrees Celsius, right? You are keeping the test tube here. How you are going to take this test tube out? By hand? Then using tongs. Tongs is just like this, right? You can, it's just like this and operate this, right? You can take the test tube from that, hold the test tube from that, tongs. So you can use it. And then you are using glasswares here. What is the risk associated with using glasswares? Cuts from the sharp objects, right? Then precaution that you can take care when handling the glass objects and keep away from the edge of the desk, right? So when you are doing these experiments, you have to be careful, right? That's why they are giving this risk assessment, right? So that's all for today, right? It's about 156, I guess. Right, now I have given some questions for you to do at home. You have to do the questions and bring it on the next class, right? So if you have any doubts now today for in today's class, just tell me, ask me any questions you have. What about Samindi? Do you have any questions?
No, teacher.
Right. So then do the questions and come, right? Then next class. Okay, you can leave for today. Right. What about you? You?
Okay,
Right.
teacher. Thank you. Have a nice day.
Okay.